Hi there. Welcome to the Proteomics section. Proteomics is the study of proteins and their functions with biological systems. Through large-scale analysis of cells, tissues, and organisms, researchers are enabled to gain insights to biological processing and diseases. In this presentation or in this lecture, we will explore some of the tools, technique used in proteomics research, and we will do it by using the code. So finding a protein in multiple databases. So searching for a protein in databases like UniProt, NCBI, PDB is a common task in proteomics research. Researchers use tools and scripts to query these databases and retrieve relevant data about protein of interest such as its sequence, structural information, functional annotation, and related literature. UniProt. UniProt is a database with protein sequence and annotations. NCBI. NCBI is a repository of protein and nucleotide sequences. PDB is the online resource with PDB structures containing different structures. So let's look at this section of code, finding the protein in multiple databases. So first of all, we will import some libraries, and after that we will define the UniProt server. Then we will add the function to send request to UniProt server. Send a request to UniProt server to fetch information about the gene P53. We are retrieving limited columns like 50 entries. Then we will convert the response into data frame using our pandas library, and then we will get the UniProt entry of P53, and after that we will get the UniProt record for specified P53 human record, and then we will print the comments and associated data with UniProt and get the list of features and then print them, and after that we will get the list of records that is present in the UniProt database. And using these, we can do this in NCBI and PDB as well. So uh, what if I tell you you can also retrieve structure of a protein using the code by Python code? So I already told this in previous lecture as well, and also you can do this by looking at the previous lecture, which will be in Bioinformatics Basics, as I think so. So I will introduce you to BioPDB. Bio.pdb is a powerful Python library that facilitates the analysis and manipulation of protein structures in PDB, protein database format. It provides a range of functions to work with protein data, such as parsing the PDB files, extracting information from the structures, computing their molecular distances, performing geometric operations. So, extract information from PDB structures in the PDB format is called parsing PDB file. So, also we can calculate molecular distances, their bonds, angles, and distance between atoms. We can also align the protein, detect similarities, and superimpose structures. So let's just move on to this section. And here is a PDB structure retrieval. So first of all, we will import the libraries, PDB, create a PDB list to retrieve the PDB files, and then we will create the PDB parser object for the PDB file, and then parse the PDB file and create the PDB structure object of each structure, and then we will print the function of the header information of PDB structure. Then we will create a function to describe the models in PDB structure, like how much models we want the how much number of residue, how much number of models. And we will describe the models, model 1, 2, 3, and then we will function to extract the FASTA sequence. And after that, we will extract FASTA sequence from the selected genes. So downloading the PDB structure 1, 2, 3, and after that, we can see if in our folder, then here are the files that we are looking for. It's 1, 2, 3, and here are the structure that we can visualize as well, and along with their sequence. Here is the sequence, and the structure shall be opened by now. So Chimera is not responding at the moment. So here you can see. So this is the protein, and that we downloaded, and we can visualize what the four loops and chains that are present in the protein. And this is the protein that you can see. So moving on, we can measure the distance between them. So for that, we have to import the module math and initialize the PDB list and PDB tracer just like before. Download the PDB structure which we already have, and then find all the zinc atom in the PDB structure, and then print the coordinates, and then define a function that will get the closest atom to get atom within a certain amount of difference you want to calculate. Calculate the number of atoms within the 4 angstrom of each zinc atom, and then calculate the number of different distances 1, 2, 4, 8, 16, and up to 20, 128 angstrom. Then measure the average time taken to execute, and then define the optimized version of get closest atom function. And we can also measure the average time taken to execute the closest alternative function as well. And we can also compare the execution time of the standard and optimized versions of function and different distances. Note that this all code is used to just extract the distances of each atom that is present in your structure, which is 1pt that we downloaded earlier. We can also identify their mass. Just like that, we have to get the protein structure. After getting it, we have to give it the unique IDs. After giving the unique IDs, we need to define a function which will be the get mass function, and then mass function will calculate the mass of atom in a chain of PDB structure, and then define a function that will get to the center of the structure, and we will that will give us the optimal weight, and then it will calculate the center of all atoms that are used there weighted by mass and each chain in PDB structure. So here you can see it has E, F, A, B, A, B, C, E, F are the chains. So here is the X, Y, Z, X mass, Y mass, and Z mass. So it's a 3D structure, so there will be 3 coordinates X, Y, Z. And you can also get the information from the file. So you can also use Praiser. So what is a Praiser? So we will import the libraries for Praiser. We will use our 1TUP file, and then we will define a function which will be praise PDB. We will open the PDB file and define the list of multiple lines, define the function of processing the multiple lines, and just like above, we will define some functions. After that, we will define a function that will process the structure type. So after processing the structure, it will gives us a result of how is our PDB file doing, like where are the coordinates of that file, uh, what type of chain is present there, what type of chain is present here, what is the score, what is the amino acid base that is present there and which is missing in the second chain, like uh, histamine is present there and glycosine is present there. So this is all what you can do. And also you can do the molecular visualization by multi-threading. You can also do threading as well. So we will import the threading module, and then we will define the function. So just like BioPDB, we will move to threading after this. So we can extract more information from PDB file as well using the BioPDB. We can also praise the model quality that we just did. We can study the protein folding as well. And we can also analyze protein and ligand interaction, which is called docking as well. So protein often combined with other molecules, which you already know, which is called docking in bioinformatics, if you know about that. And then we can also calculate the bond length, angle changes, their spatial relationship that we just discussed. And also we can identify some conserved protein regions by using the BioPDB, explore the structural relationship, and also we can align some proteins. Here you can see we are moving to the threading option. So what is threading? So it is a type of biology bioinformatics protein structure identification, just like homology. It is threading. So what threading basically do, it gives you the protein threads, how many threads are present in the protein. So here you can see all this code is used to just to get the threading history of a protein. So it is not very important code. You can just check it by yourself if you want to. We will see how more information we can get from a protein structure. So let's just say we want to get our angle length and bond angle and bond types that we were previously discussing. We want to calculate the bond length, angle changes, and spatial relationship. So for that, we will import some modules. After that, we will initialize our tracer and PDB list. Then we will initialize the default directories, and then we will loop through all the residues that are present in the protein and print the statistics of atom per count of the residue type as well. So then we will print the statics. After that, we will define a function to calculate the bounding box, initialize a dictionary that will store the bounding boxes of each chain, and then loop through all chains and calculating their bonding length. Calculate and print the bounding box of entire structure, and then we will use the MATLAB to 3D plot and visualize the 3D structure and its chain. So here you are seeing, here is the 3D structure of our protein, and here you are seeing that they are constructing at a spatial length. So if I can save as copy to clipboard, if I can open this image, oh, sorry about that. If we can look at this image, it is the representation of your protein bond and angle length, and it is also a 3D structure. You can visualize it, but it is not happening right now in my code. So before moving on further, you should know that proteins are 3D structures. You can 3D theoretically you can use them. You can see their structure. I should move to a 3D structure. So here you are seeing it is a 3D structure of a molecule with a DNA attached to it. I just recently bonded it. So if I can just and make it more like which can, yes, if you can see it right now, it should be a white screen. So now you can see that the protein is attached to uh, a DNA. If you can see a DNA structure is added to a three different type of proteins. So you can animate these proteins. You can visualize them interactively by using a important software which is called Pfam. So you can use that as a command line tool as well. So for that, what you have to do, you can animate this is animate it with using the Pfam. So Pfam is a widely used visualization tool in proteomics that allows researchers to create stunning 3D visualization of protein structure. Using Pfam with combination to BioPDB, researcher can animate protein movement, explore molecular dynamics, visualize protein-protein interaction, visualize protein motion. Animation is a powerful feature of Pfam that allows researcher to visualize the protein dynamics. And you can also explore the protein-protein interactions. Pfam enables researcher to visualize interaction between two more. You can also use 5 to 6 proteins for allowing a better understanding of protein function. But you can also use this to visually see how a ligand is being attached to your protein. For example, a protein ligand interaction can be stunningly steady by using the animation protocol by Pymol. So, and let's just do that. So, by using this, you have to use a lot of resources. First, we will import the Pymol module from the Pymol, and then we will launch it in our code. And after that, we will fetch the PDB file which is present in our directory, disable all the representations of our structure, show the surface of structure which will be the A, chains of our structure which will be A, B and C, and also the cartoon representation of our E and F chain. Create a scene SO and store the current view, show the cartoon representation and hide the surface representation, create a new scene and then hide all the other cartoonish representation, and then just set the view and create frames. So after that, it will gives us a movie type of representation of our protein with some images in it. So what will be the movie that will be shown to our terminal? So here you can see there is a protein that is attached. So what type of protein this is? So I just recently showed you a protein that has a DNA attached to it, just like this. Here is the hydrophobicity surface of your protein, and the DNA, which is a ligand, is now being attached to it. So if I just close this and open this structure again, and let's just say if we want to make it look like that. So we will go to this and just create a hydrophobicity. So it is not running right now because it is being used in the code, but you can see it is a similar structure like that. So let's just movie. So here you are seeing it is visually like a movie. It is now being zoomed in to see the interaction. So it is a very good thing for using in your protein and ligand interaction. So you can use this for your own proteins. You can visually and stunningly create movies like this for the proteins for your research papers to show it on your presentation and things like this. So here you can see all the ligand and protein interactions being showed. And here is the structure of that protein. You can make movies by this by using the visual and the code that I have prepared. So praising the mzIF file. So what are the mzIF files? So these files are the advanced format. So researcher often encounters protein structure format in macromolecular crystallographic information file format. So BioPDB supports praising of mzIF files as well. So it will give you access to the broad range of structural data and facilitating more comprehensive protein analysis. So it contains more detailed information about protein structure than PDB, including the atomic positions, experimental data, and also it is easier for the analysis because it contains diverse amount of data as well. So for that, we can just see this code is used for to trace a structural file which is in the mmcf file. So here you can see there are A, B, C, D, E, F chains that contain this number of residues, this number of amino acids. E contains 1100 amino acids. F contains 1101 amino acids. A chain contains 94 amino acids. This is all the information that you can get in your uh, mmcf file. And there is an audit entry, there is audit confirmation, how much files these, is there zinc, iron, and other extensively monopoly atoms that are present in it. So that is it for the lecture of proteomics. I hope you have understood that, and that's quite complex things you have to go through if you want to study the proteomics section. But all the codes are available. You can just use it for your own files, retrieve the information like you want to, and that's pretty much it. Thank you so much for your attention.